Hello friends, today we will discuss structure of RBC and their functions. Now the red blood corpuscles or RBC, they are also called erythrocytes. They are the major cellular elements of the blood. Out of 100 trillion total cells in the body, the RBC counts for as much as 25 trillion. The normal mature erythrocyte is circular, disc-shaped. It is biconcave. It looks like this, and it is non-nucleated, as when the erythrocyte matures, the nucleus is disintegrated and the hemoglobin is accommodated in the whole cell. Now, regarding the shape, as just mentioned, it is biconcave. The size is 7 to 8 micrometer, μm. The thickness is 2 μm at the periphery here, where it is thicker, and it is only 1 μm at the center, where it is pinched like this. When the RBC matures, it becomes biconcave, as if if you take a small balloon and you press it with your finger and the thumb, then it becomes thinner at the center and thicker on the margins. So this is what happens when RBC gets mature. This arrangement or this shape of RBC facilitates displacement of hemoglobin towards the periphery, so that maximum hemoglobin is in contact with the air, so that maximum oxygen can be absorbed in RBC. Then surface area of RBC is about 140 square micrometer. The volume of RBC is about 87 cube micrometers. Now, the RBC count. The normal RBC count in case of male is 5 million per cubic millimeter, whereas it is little less in case of female. That comes about 4.5 million per cubic millimeter. This difference in RBC count is mainly due to the blood loss that takes place during menses. In case of infants, the count of RBC is about 6 to 7 million per cubic millimeter. In case of fetus, it is even more, that is 7.8 million per cubic millimeter. Now, if the count of RBC decreases, that means the amount of hemoglobin will be decreased, and that is a condition which is called as anemia. Another term opposite to it is polycythemia. Poly means more. Increase in number of RBC count is called polycythemia. In case the RBCs are produced more in number, then their count will increase, and that condition is called as polycythemia. Now, variation in RBC count. As you can see, the RBC count varies by 5% during the day that we call Diurnal variation. The minimum number is observed in the morning time when the metabolism is at the lowest level. So, the number of RBC are minimum, whereas the RBC count is maximum during the day, in the evening, when the metabolic condition is little raised. Then altitude. As we move on to higher altitudes, gradually as we move, the oxygen amount in the air decreases. And due to that decrease in the amount of oxygen, two physiological changes occurs in our body. Number one is the respiratory rate increases to cope up with the deficiency of oxygen in air. And number two, the number of RBCs increases to cope up with the deficiency of oxygen in the blood, so that sufficient amount of oxygen is delivered to the tissue. Then in case of exercise, when one is doing exercise, his metabolic rate increases, and in that condition, he needs more amount of oxygen to maintain the metabolic rate. And therefore, during exercise, the number of RBC increases temporarily. Now, another factor for increase in the RBC count is the oxygen tension. When oxygen tension of arterial blood is lowered, then RBC count increases. Then another important factor is the temperature. When the body temperature increases, the metabolic rate increases, and the need of oxygen in the tissue increases, and to cope up with that need of oxygen, RBC count increases. Another physiological condition in which the RBC count increases is the state of excitement. When a person is in the state of excitement or stress, adrenaline is released or sympathetic stimulation occurs that increases the metabolic rate to cope up with the excitement and stress conditions, and in that, number of RBC increases temporarily. Now, certain medical conditions in which the RBC count increases. Number one is in case of heart failure, when there is some problem in the heart, if heart is under the stress or a heart attack occurs, then sufficient amount of blood is not supplied to different organs. That leads to hypoxia. And in case of hypoxia, always the kidney secretes a proteinaceous hormone called hemopoietin, which increases erythropoiesis in the bone marrow. That is how the number of RBC increases. Another is same as in case of congenital heart diseases. In case of children with congenital heart disease also, sufficient amount of blood is not supplied. Then in case of polycythemia, polycythemia vera or blood cancer is a condition in which the hemopoiesis in bone marrow increases abnormally, thereby increasing the number of all type of blood cells, including RBCs. Another condition is that of kidney tumors. You know, in kidney, between the renal tubules, there is a tissue, interstitial tissue called hemopoietic tissue. If there is a tumor in kidney, especially of hemopoietic tissue, then it will secrete more amount of erythropoietin, which is a hemopoietic stimulating factor. Then in that case, the number of RBCs produced will be more abnormally, so it will increase the RBC count. Now, certain medical conditions in which the RBC count increase is observed. One is in the case of lung diseases, you know, when there is some disease in respiratory organ, the less amount of oxygen is diffused from the lungs into the blood. So, tissue hypoxia occurs that increases the RBC count. Like in case of emphysema, pulmonary fibrosis, COPD. So, in all these medical conditions, RBC count increases. Then CO poisoning. As you know, the hemoglobin has got 200 times more affinity to carbon monoxide. And the hemoglobin combines with carbon monoxide, forming a compound, carbon monooxyhemoglobin, which is a stable compound. So, that amount of hemoglobin is not available for forming oxyhemoglobin with oxygen. So, there is a deficiency of oxygen, and in that case also the RBC count increases. Now, there are certain lifestyle factors in which the RBC count increases. One of that is smoking. In smoking, you know, carbon monoxide is produced, as just mentioned. Because of that, the count increases. When one goes on high altitude, this is a very common phenomena. When a tourist goes on to high altitude, his RBC count increases temporarily, and in people who live there, their RBC count is always higher than those people which lives in the plain. Now, there are certain drugs like anabolic steroids, testosterone, erythropoietin, and other performance enhancing drugs. As you know, as in case of sports and other activities, when an athlete or any other person, a wrestler, wants to improve his performance, he needs more amount of oxygen to generate more energy, and for that he needs more number of RBCs. Therefore, certain drugs are taken, although it is illegal these days, but these drugs increases the production of RBCs. Now, symptoms of low RBC count. If the RBC count decreases, then there is weakness because of the factor that oxygen is not available to maintain the metabolic rate and production of energy. Then there is a fatigue. So, if oxygen is not available, some anaerobic respiration will occur that will cause the accumulation of lactic acid that causes fatigue. Then paleness of skin. The redness of skin is due to the red pigment of blood, that is hemoglobin. So, if it is less, then skin becomes pale. Then rapid heartbeat to cope up with this situation, to increase the cardiac output so that the tissue gets sufficient amount of oxygen. Then symptoms of high RBC count. If there is a high RBC count, then what are the symptoms? So number one is headache, dizziness, and there may be some problem in the vision. Now, structural abnormalities. A normal RBC with disc-like shape, biconcave shape, is called normocyte. It is biconcave, anucleated, and disc-like. But there are variations. There are abnormal variations in the shape of RBC, like one is macrocyte. That means the RBC are larger, that is more than 94 cubic micrometer. They are found in the case of megaloblastic anemia. As you know, when the RBC matures from the erythroblast stage, its size regularly decreases, its nucleus degenerates to accommodate more amount of hemoglobin. But if the vitamins like folic acid, vitamin B12 is not sufficient, then those stem cells of RBCs which matures into RBC, they remain larger in size and nucleated. Such RBCs are called macrocyte, because such RBCs do not have that sufficient amount of hemoglobin, number one. Number two, their membrane is not that much flexible. So when they pass through the spleen, because of their larger size and the fragile cell membrane, they are destroyed. And such anemia caused by the larger RBCs is called either macrocytic anemia or megaloblastic anemia. Now, the opposite case, that is microcyte, smaller RBCs less than 80 cubic micrometer, occurs in case of iron deficiency anemia. The size of the RBCs is reduced than the normal, and the hemoglobin content is also not up to the standard. It is less than the standard value. So it is called as microcytic anemia, case of iron deficiency. Now here in this picture, you can see various types of abnormalities in the structure of RBC. In the first case here, you see that these are microcytic hypochromic anemia. That means their size is less than normal and they contain less hemoglobin in them. So in that case, it is not able to provide sufficient oxygen to the tissue, and therefore it is called anemia. And as the cells are smaller, it is called as microcytic anemia. And as these cells contain less amount of hemoglobin, it is called hypochromic anemia. Now in the second condition, it is called megaloblastic anemia. As I just described, the RBC remains larger, they remain nucleated because of the deficiency of vitamin B12 and folic acid. And when such RBCs passes through the spleen, they are broken, causing anemic condition. So this is called as megaloblastic anemia. Now, here is another case where the shape of RBC is affected, that is sickle cell anemia. In this case, it is a hereditary disease in which the beta chain of hemoglobin is affected, and there is a shift in the amino acid, and that causes defect in the hemoglobin as well as the shape of the cell is also damaged. They become sickle shaped. So this is called sickle cell anemia. And then erythroblastosis fetalis. Now this happens when there is a Rh incompatibility between mother and the fetus. You know, if the mother is Rh negative and if the fetus is Rh positive, and if the mother already have Rh antibodies due to earlier transfusion of blood or due to first pregnancy of Rh positive fetus, maybe in that case the antibodies from the mother crosses the placental barrier and reaches into the fetal circulation, thereby it causing the hemolysis of the fetal RBCs. Those RBCs becomes blue in color, they are destroyed. So this is also sometimes called as blue baby. In later lectures, we will discuss this issue in detail. Now, if there is a variation in size, it is called anisocytosis. Like spherocytosis, if the RBCs becomes spherical in shape, it is a hereditary disease called hereditary spherocytosis. Similarly, if the RBCs becomes elliptical in shape, it is called as hereditary elliptocytosis. It occurs in case of thalassemia and iron deficiency anemia. Now, any deviation from the normal shape of RBC, such RBC is called poikilocytes. If they become spherical, if they become elliptical, then all these variations, if they become sickle shape, all such variations are covered in a common term called poikilocytosis. Now, characteristics of RBC. The first and foremost characteristic is their greater number and smaller size. As you know, the number of RBC is the maximum in our body. Secondly, their size is small. So, in blood, larger number of RBCs are accumulated. And as you know that the RBC contain hemoglobin, and hemoglobin carries oxygen, 4 molecules of oxygen. So, maximum amount of hemoglobin is contained in RBC because of their shape and their number and their size. Second is the absence of nucleus. You know, during the maturation of RBC, in the late normoblast stage, when it converts into reticulocyte stage, the nucleus becomes smaller, it becomes condensed, and ultimately the nucleus and other cytoplasmic organelles like mitochondria, Golgi and others, they disappear totally so as to provide maximum amount of hemoglobin. Then this absence of nucleus also provides a space to facilitate more amount of hemoglobin, and the RBC becomes biconcave in shape. Now, another important characteristic of RBC is their biconcave shape. This biconcave shape facilitates the movement of RBCs, and they can withstand the pressure when they passes through the tortuous network of capillaries through the organs. Say for example, if you have to pass from a barbed wire boundary, then you have to pass like this, you have to pass like this from that barbed boundary. And similarly, what happens when there is a tortuous blood vessels, the RBC, they passes like that. So they can easily withstand the pressure. Now, due to biconcave shape, hemoglobin is distributed in thin layer. This facilitates quick exchange of gases. When the RBC becomes in this shape, what happens? The hemoglobin is distributed more in this area like this. It is distributed throughout the periphery, which is now having greater surface area than it would have been in a circular shape, so that the hemoglobin gets maximum surface area to come in contact with the oxygen to form oxyhemoglobin. Now, the volume of RBC can increase due to carbon dioxide tension. As you know, when the blood passes through the tissue, carbon dioxide comes in blood from the capillaries. That carbon dioxide enters into the RBC, thereby increasing the volume of RBC. It appears in the case of acidosis, anoxia. The volume of RBC is slightly increased in venous blood also. As you know, when CO2 enters into RBC, it forms carbonic acid. And this carbonic acid dissociates into HCO3 ions. So, during this phenomena, the volume of RBC increases. Therefore, the volume of RBC in venous blood is more than that of arterial blood. Another is the blood volume. Now, here, due to chloride shift in RBC in the venous blood, the volume of cell increases, as I just mentioned. Because of this, the volume of cell increases. This increases due to increase in the osmotic pressure and drawing more water into the cells. This allows easy folding of RBC in the capillaries. So, all these things are mixed. When HCO3 ions comes out of RBC, then one chloride ion enters into it. So, one HCO3 ion is excluded from the RBC in lieu of a chloride ion which is taken in. And in this process, the volume of RBC increases. It helps the RBC to withstand the osmotic pressure changes, and it can pass easily from the capillaries of the spleen. Now, another factor, we have just mentioned that RBCs are anucleated. During the process of maturation, the nucleus disintegrate, mitochondria, ribosomes, RNA, DNA, all these are disintegrated, and RBC is completely occupied by the hemoglobin. So, therefore, hemoglobin synthesis of proteins and lipids does not occur in mature RBC because it does not have nucleus, it does not have mitochondria where heme synthesis occurs, it does not have ribosomes where protein synthesis occurs, it does not have DNA to command all these synthetic processes. So, mature RBC mainly depend upon glucose, which they obtain from plasma for active transport of calcium and potassium ions. Glycogen is absent. That is the main point, which decides the metabolic capabilities, the metabolic characteristics of RBC, that it depends totally on glucose. Glycogen is absent. Now, metabolism of RBC. Glucose is metabolized through EM glycolytic pathway and hexose monophosphate shunt, that is HMP shunt. Now, the important thing is Krebs cycle, that is TCA cycle, is absent. That means in RBC, aerobic respiration does not occur. It performs metabolic activity by only anaerobic method. The ATP required for transport mechanism of RBC, RBC has to pump sodium out and potassium in. For this process, a continuous pumping mechanism is needed, which requires energy, and it requires energy in terms of ATP. Therefore, longevity of RBC, because it does not have any synthetic activity, so it totally depends upon whatever enzymes it has already got. So, the longevity of RBC depends upon the metabolic process and production of RBC. That is why we see that the lifespan of RBC is limited, that is 120 days. Now, here in this picture, you can see the RBC, the source of energy. You can see it draws glucose from the blood plasma, that is metabolite into lactate, that produces 2 molecules of ATP. This provides energy for the RBC. Now, these 2 molecules are used for the maintenance of membrane, the maintenance of volume, maintenance of shape, and maintenance of flexibility. Here you can say 1 molecule ATP is used for this pumping mechanism, for pumping 3 sodium ions outside and 2 potassium ions inside. So, this is how the RBC uses glucose for maintaining the metabolism and production of ATP. Here is another picture which depicts in detail the total EM glycolytic pathway. Here you can see the ATP is used and converted into ADP. You know, this is an anaerobic respiration in which 4 ATP are produced, 2 of which are used in the process and 2 ATP remains for use of RBC. So here you can see the glucose is converted into lactate and the ADP is converted into ATP. Yeah, this adequate ATP level is needed for what? It is needed for survival of RBC. It is needed for hemoglobin functions. It needs for the membrane integrity, shape, RBC volume, and so on. Now, as I mentioned just now, that RBC generate energy by anaerobic breakdown of glucose. Now, coming on to the lifespan of RBC. The lifespan of RBC is about 4 months, that is 120 days. It is said that the enzymes required for maintenance of shape of RBC is limited for only this period. Older RBC changes their shape and size and becomes brittle. Such RBCs are called poikilocytes, as I mentioned earlier. Their shape is changed, their membrane gets brittled, and then when they passes through the spleen, they are broken. Now, the fragments of RBC, when the RBC gets fragmented in the spleen, then those fragments are engulfed by the reticuloendothelial cells of spleen, liver, and the senile RBCs are also, senile means old RBCs, are also destroyed and they are sent to the macrophage cells which digest them, and hemoglobin is released. Then this hemoglobin is degraded. This is an important point here. Hemoglobin is degraded into verdohemoglobin or choleglobin, and it is circulated in the blood. Now, these verdohemoglobin, when it reaches in the blood circulation, it is again broken into protein and heme. Protein is finally broken into amino acids, which are used as amino acid pool in the blood for synthesis of certain enzymes and proteinaceous hormones and other structural proteins. Now, the second component of hemoglobin is the heme. Heme is stored as ferritin and hemosiderin, which is used in synthesis of hemoglobin. So, this heme portion, which contains iron, is again reused for synthesis of hemoglobin. Now, some amount of heme remains in the blood. What happens to that? It is converted into a yellow pigment called bilirubin and a green pigment called biliverdin. These two pigments, when reaches to the liver, then they are separated from the blood as bile pigments. So, that bile is finally eliminated from the liver in the form of bile juice, and it is poured in the small intestine with the help of bile duct into duodenum. Now here in the intestine, these bile pigments are converted into stercobilinogen, which is excreted through the feces as a yellow-brown pigment called stercobilin. The color of the fecal matter, that is brownish yellow, is due to this degraded bile pigment present in the feces. Now, some amount of bile pigments is reabsorbed in the blood, and whatever is present in the blood, that is taken away in the circulation and finally reaches in the kidney, where it is eliminated as urobilinogen in the urine, which is responsible for the yellow color of urine. Now, this whole process you can see in this picture, when hemoglobin is degraded into choleglobin, which degraded into hemosiderin and ferritin, which is again used for synthesis of hemoglobin in the liver. Then the pigments are converted into bilirubin and biliverdin. These two pigments are separated in the liver as bile juice, and this bile juice is sent to the small intestine, that is duodenum, and from duodenum it is passed into the large intestine, where by bacterial action it is converted into stercobilin, and stercobilinogen first, then stercobilin, and this is responsible for the brown color of feces. Some amount of urobilinogen reaches into the kidney, where it is filtered and it appears in the urine as urobilinogen or urobilin, which is responsible for yellow color of urine. Now, functions of RBC. The main function of RBC is in the respiration. As we know that the RBC mainly contains hemoglobin, and near about 640 million hemoglobin molecules are present in single RBC, and each hemoglobin molecule is capable of carrying 4 molecules of oxygen. So that is how the role of RBC is there in respiration. Secondly, it also carries carbon dioxide from the tissue to the lungs. Then the RBCs are helpful in maintaining ionic balance, as I just mentioned that when the carbon dioxide enters in the RBC, then bicarbonate ions are released from there in lieu of a chloride ion called chloride shift, and by that the ionic balance is maintained in the blood. Now, the same thing here, by carbonic ions and by hemoglobin, that is a protein, the acid-base balance is maintained in the blood and other inter and intracellular fluids. Now, RBC plays an important role in maintaining the viscosity of blood. The viscosity of blood is important for maintenance of the volume of blood. As you know, the RBC contributes nearly about 50% to the viscosity of blood. Now, hematocrit or packed cell volume. When the whole blood It is the percentage of RBC in whole blood. It can also be defined as the amount of packed RBCs following centrifugation. When the whole blood is subjected to centrifugation, then by centrifugal force, the heavier RBCs settles down at the bottom of centrifuge tube, and the amount of that packed cell is called as packed cell volume or hematocrit value. It is a reliable index of RBC population in the blood, and it is used for detection of various kinds of anemias. Now coming on to the values of hematocrit, that is packed cell volume. In case of male, it is 47%. In case of female, it is little less, that is 42%. And in both the cases, there is a variation of plus minus 5%. High hematocrit values are found in case of polycythemia. That means when the RBC count increases more than the normal. Similarly, lower values of hematocrit are formed in case of anemias. Now, another parameter used in the diagnosis of blood is the ESR value. Hematocrit value was the sedimentation rate of RBC after centrifugation. Here, the sedimentation of RBCs without centrifugation. The RBCs have a tendency to pile one on the other like this. Forming Rolex. So when blood is allowed to settle in a tube, RBCs settle by gravity. The rate of sedimentation is called ESR value. Now, the ESR values, here are normal values. In case of Winthrop tube, males 0 to 9 mm per hour, females 0 to 20 mm per hour. In Westrogen tube, males 3 to 5 mm per hour and females 12 mm per hour. ESR is high in case of newborn, infants, and in case of female. ESR increases in pregnancy, during menses, and at the time of parturition, that is childbirth. Now, abnormal values of RBC, what it indicates. ESR increases in certain pathological conditions like tuberculosis, chronic inflammations like arthritis, cellulitis, malignancies, that is cancers, chronic infections like tonsillitis, and different types of anemias. ESR decreases in case of polycythemia when the number of RBC abnormally increases, and in case of sickle cell anemia when the RBCs becomes abnormal, having a shape like sickle. Now, here you can see a recap of the erythropoiesis. Here are proerythroblast, which is converted into basophil erythroblast, that is early normoblast, which are then converted into polychromophil erythroblast. You can see the reduction of size from here to here and change in the nature of cytoplasm and nucleus. When it comes to orthochromatic erythroblast, that is late normoblast stage, here formation of hemoglobin starts, the nucleus shrinks, and then in the next stage, reticulocyte, the nucleus is finally disintegrated and the RBCs matures as a biconcave disc-like corpuscles completely filled with hemoglobin. Now, you can see here the phenomena of erythropoiesis, how it is affected by the hormone called erythropoietin. You know, when there is a decrease in oxygen, then the erythropoietin is released from the hemopoietic tissue of kidney. It regulates the production of RBCs, and when the oxygen level becomes normal, then the production of erythropoietin decreases, thereby reducing the production of RBCs in bone marrow. Now, a little idea about how the RBCs are counted. You know, the instrument needed for this is called hemocytometer. It contains a RBC pipette, RBC counting chamber, that is called as Neubauer chamber, a compound microscope, and chemicals which are called as RBC diluting fluid. One is the Hayman's fluid, another is Gower's isotonic solution. Now, here is the composition just for your reference of Hayman's saline and Gower's saline, which is used for dilution of the blood sample. Here you can see the picture of the complete instrument. It includes a pipette for drawing the blood sample. It is identified by a red bead present in this part. The blood is taken up to this 0.5 mark, then the diluting fluid is sucked with the help of suction tube up to 101 mark. Then it is properly mixed by rotating this pipette, and then this Neubauer chamber is used for counting of the RBCs. The fluid, first few drops are discarded, then this blood with diluting fluid is poured into this counting chamber, and then the number of RBCs counted on the four corners and in the center one, and then total number of RBC counted is multiplied by 200 into 10 into 5, that is dilution factor, depth of the cell, and so on. So it comes about 5 million per cubic millimeter. Here is the same procedure as I have just narrated. About 2 readings are taken, then their average is taken for final calculation. A magnification lens of 40x is used for counting the RBCs. Then here, whatever number of RBCs are counted, they are multiplied with a factor, this one, $10,000, and then the final number of RBC is reported as millions per cubic millimeter. That's all for today, friends. Thank you very much.